The overall goal of this experiment is to investigate the gold sorption behavior by S-layer proteins of the presented bacterial strain from an ecological and technological point of view.
These methods can answer key questions in environmental-related science, such as fields of sorption processes and removal and recovery of metals. The main advantage of these techniques are that you can detect metal sorption and nanoparticle adsorption in a nanoscale range. By using QCM-D, it is possible to detect this in a real-time measurement.
To begin this procedure, equip the fluid cells with sensor dummies. Pump at least 20 milliliters per module of a 2% alkaline liquid cleansing agent in ultrapure water through the QCM-D and tube system. Following this, pump a 5-fold volume per module of ultrapure water through the system with a flow rate up to 300 microliters per minute per the manufacturer's protocol. Clean the silicon dioxide sensors outside the flow modules by incubation in 2% sodium dodecyl sulfate solution for at least 20 minutes. Then, rinse the sensors several times with ultrapure water. After drying the crystals with filtered compressed air, place them in an ozone cleaning chamber for 20 minutes. Following this, repeat the cleaning procedure twice to remove all organic contents. To remove bound metals from the sensor surface, rinse the sensors